Can somebody confirm that you hear me?
I can
I
hear
can
you.
hear you.
Thank you. For some reason, my computer is really slow. So give me a moment here. Before we begin, are there any questions?
Can you please just confirm what chapters this first quiz is going to be on? Is it going to be 1 through 3 and 10?
Not entirely all of three. Let me get into that. I'll show you the screen in a minute here, but like I said, my computer is working slow for some reason. I've even shut some programs down, but I'll probably need to reboot it. Maybe I can do that between the lecture and the lab. All right, let me share my screen and then I'll answer that question. So in the, I don't know why that even comes up. Um, in the syllabus, in the schedule of the classes, I've got what the exam will be covering. And this exam, this Wednesday, which will be the 21st, if I'm not mistaken, will be quiz one. It'll cover chapter 1, chapter 10, only chapter 10, what we covered, the start of chapter 2. So everything that we covered so far in chapter 2, as well as lab 0, 1, and 2, will be on the first quiz. Any question about any of that? All right, today is January 20th, and there will be a lab today. We will continue talking about chapter 2. We're actually supposed to end chapter 2 today. I think we're behind schedule, and we probably will not successfully end chapter 2 today. What else? Oh, I was going to talk a little bit about the grading. I'll hold off and talk about the grading in the lab because we're a little behind schedule. Any questions about anything? All right, so I have on the lecture that we had talked about organic compounds and that they can be made larger by a dehydration synthesis reaction, which is also called the condensation reaction, where water comes out, making a larger molecule, in this case a polymer, from the makeup of the smaller molecules, the monomers. But we can also break down a polymer or a larger molecule by adding water. This is called a hydrolysis reaction, and it will split polymers into monomers. Looking at that in a different form, here we have a monomer, and this is going to actually join with a smaller polymer, hydrogen coming off the polymer, OH coming off the monomer, forming water, and then the monomer will be bound directly to the polymer. So that's dehydration synthesis. And then hydrolysis reaction will be the reverse, where you add water, which will then break apart with the OH group coming onto the monomer, making a monomer come off the polymer, and then we'll have a smaller polymer with the H of the water joining on the polymer. Any question about any of that? All right, if not, let's talk about the first of the biological molecules, carbohydrates. These include sugars, starches, and other complex carbohydrates. Carbohydrates are an important energy source. We use them for cell structure. They're used in making nucleic acids. Carbohydrates always consist of carbon, hydrogen, and oxygen. But they're not just any combination of carbon, hydrogen, and oxygen. They always are of the general formula. For every one carbon, there's two hydrogens and one oxygen. And for a carbohydrate, you just have this CH2O, and then you repeat that n times to make the carbohydrate. For example, glucose, as well as fructose for that matter, is going to be CH2O with an N of 6, meaning glucose is C6H12O6. Any question about any of that? Carbohydrates include the simple sugars, and you just string the simple sugars together to make the larger carbohydrates. You can classify carbohydrates into three major groups based on the size. A monosaccharide, also called the simple sugar, a disaccharide, these are just two monosaccharides linked together, and then a polysaccharide, which literally means many saccharides or many monosaccharides linked together to make the polysaccharide. Any question about any of that? Starch is a simple polysaccharide, and then the simple sugars, glucose and fructose, are examples of monosaccharides. If you want to know, sucrose, shown here, is a disaccharide. Monosaccharides are always simple sugars. They always contain between 3 and 7 carbon atoms, but usually most monosaccharides have 5 or 6 carbons. With glucose and fructose both have 6. Ribose and deoxyribose always have 5 carbons. The simple sugars are sweet and water soluble. All the monosaccharides are sweet and water soluble. The disaccharides are formed by joining two monosaccharides in a dehydration synthesis. Let me blow this up so you can see it. So here we have glucose and fructose. Water comes out in dehydration synthesis, and that will link the oxygen of fructose directly to the carbon here of glucose. Right there. Or wait a minute. It's right here. Sorry, right here. And that's how glucose and fructose make sucrose. If you don't know, sucrose is table sugar. So the sugar you normally have on your table is called sucrose. Sorry, I don't know why that's not going down. It should go down there. Oh well. Any questions about how you make a disaccharide or how you can change a disaccharide like sucrose by adding water, which is shown there? The water will split apart, the H joining the fructose and the OH joining the glucose to form glucose and fructose in a hydrolysis reaction going this way. Any question about any of that? All right, let's talk about the polysaccharides. Polysaccharides consist of many monosaccharides or simple sugars. You can have straight or branched chains in a polysaccharide. Some examples of polysaccharide include starch, glycogen, cellulose. All of these, starch, glycogen, and cellulose, are simply chains of glucose linked together, one after the other, to form either the starch, the glycogen, or the cellulose. There's also chitin, but we're not going to talk about it. Let me end this and then you can see this a little better. If we don't have that there. Here's one of the simplest polysaccharide starch. It's usually just glucose hooked together one after the other in a simple linear chain. At least most of the time, it's a simple linear chain. One after the other, glucose making starch. And here are some starchy foods. Everybody knows bread, cereals, and pasta. Rice and potatoes are starchy foods. But you probably didn't know beans and chestnuts are also considered starchy foods. Beans are about 1/3 carbohydrate, 1/3 protein, 1/3 fat. And chestnuts are something similar. I don't know the exact makeup of chestnuts. Any question about any of this? Here we're looking at starch. Like I said, it's mostly a single linear chain, straight chain, although it can occasionally branch, not very often. Glycogen is the same, simply glucose one after the other in a chain, but glycogen is a branched chain, much more branched than starch. Cellulose, let me blow this up really, really big here so you can see it. Cellulose is also glucose hooked together one after the other in a chain. And there is the first chain with the dark purple line being the covalent bond hooking this sugar to that sugar, that sugar to that sugar. Cellulose is different in that we have rows of chains of glucose coming next to each other. And these rows of glucose hydrogen bond with the neighboring glucose. So here's a glucose here, hydrogen bonding to this one. This glucose is hydrogen bonding, obviously, to that glucose. It's also hydrogen bonding to this glucose. So these dashed lines are hydrogen bonds. And as you can see, cellulose has a lot of hydrogen bonds between the glucose molecules. That makes glucose difficult to digest. And people, as you know, cannot digest cellulose. Cows by themselves cannot digest cellulose either, but they have four stomachs, and in their stomachs they have microorganisms which can digest cellulose. And when the microbe digests the cellulose, it makes the digested cellulose available to the cow to then digest it. And that's how a cow can actually digest grass or other matter that has cellulose in it. So a cow can digest cellulose, not directly, but because it has microbes in its stomach, which can digest the cellulose. Any question about any of that? And if you really want to know, the cow has a protus in its stomach, which can't digest the cellulose, but the protus has microbes in it that can digest the cellulose. So the bacteria and the archaea in the protus digest the cellulose, make the cellulose available to the protus, and then the protus makes the cellulose available to the partially digested cellulose to the cow. And that's how the cow can digest cellulose. Any question about any of that? And no, you don't need to know this slide. You don't need to know about cows and how they digest cellulose. We will talk a little bit about this one here, the peptidoglycan. It does have some sugars in it. It's largely a sugar, and then there's chains of them, but it also has peptides linking the sugar chains together. And we'll talk about that later in the term, maybe even later today, either today or tomorrow, or not tomorrow, but Thursday. We'll talk about cellulose, not cellulose, peptidoglycan. Any question about any of that? So, like I said, you don't need to memorize this chart of the complex carbohydrates. I'm just showing you some of them. Any question about the carbohydrates? If not, let's move on to the second of the biological molecules, the lipids. Lipids consist of carbon, hydrogen, and oxygen, but they do not have very much oxygen. I'll show you, well, I guess I can show you now. Here's a very simple lipid, a fatty acid. I can blow that up, move that out of the way. You can see that this very simple lipid, the fatty acid, is mostly a carbon-hydrogen chain. All of this part of the molecule, carbons and hydrogen. There's only two lipids, oxygens, on this lipid, right there and right there. And it'll lose this oxygen actually when this combines with something else. We'll talk about that in a minute. So the point of the matter is lipids do contain oxygen, but not very much. They're mostly carbon and hydrogen. Come on. Lipids are mostly nonpolar and insoluble in water. There is one exception. We'll talk about that exception. That means they're hydrophobic. They do not like to be around water, and they will not, for the most part, they will not dissolve in water. And you should know that because when you mix vinegar and water together to make your salad dressing, the oil always separates from the vinegar, which is in mostly water. I think regular vinegar is only 5% acetic acid and 95% water, just to let you know. Lipids are structural and energy storage molecules. They are the primary components of cell membranes. It's a phospholipid. It's the primary molecule in the cell membrane or any membrane for that matter. We can separate lipids into three groups: the simple lipids, complex lipids, and steroids. Any question about lipids? All right, let's take a look at a fatty acid, the simplest form of a lipid. It's also a major component of many other lipids. So like a fat, which is a lipid, it's made up of fatty acids and then one glycerol molecule, and we'll talk about that in just a minute. A fatty acid consists of a chain of carbons held together one after the other, holding the different atoms together, meaning all the hydrogens here are linked together by the carbon-carbon backbone. The same with that oxygen here and this OH group there. Fatty acids are mostly a, a long chain of or semi-long chain of carbon and hydrogen units, meaning this part of the molecule here. Which is this part of the molecule is only a hydrocarbon, only carbon and hydrogen. They do have a COOH group on this end, one end of the molecule. Other locations are just hydrogen bound to a carbon. We have two types of fatty acids: a saturated fatty acid, shown here, where all of the carbon links are a single carbon bond. So this is a saturated fatty acid. We also have an unsaturated fatty acid, which has a double carbon bond linking the carbons together. Wherever there's a double carbon bond, we have a kink at that location where the molecule will be bent right here. And I'll show you that right here. So this is a simple fatty acid right there, that one too. And these are saturated fatty acids because they're simply single carbon bonds holding the carbons together in the fatty acid. This is actually a fat composed of three fatty acids and then one glycerol molecule. The glycerol molecule shown here combines with the fatty acid shown here. The OH group comes off of the fatty acid, And that will link this carbon directly to this oxygen on the glycerol. The hydrogen comes off the glycerol. For triglycerides or most fats, you have a glycerol molecule combined to three fatty acids shown like here, where the glycerol binds to a fatty acid at each location of the oxygens here. So a triglyceride is made up of three fatty acids and one glycerol. Why it's called a triglyceride? Because there's three fatty acids. Any question about any of that? All right, let me blow this up so I can show it better. Come on. Oops, didn't mean to do that. Trying to move this out of the way. This is a saturated fatty acid on this fatty acid group here, meaning it's held together only by single carbon bonds. Here we have an unsaturated fatty acid that has one or more double carbon bond holding the carbons together. So you only need one to be called unsaturated fatty acid. Also, when we're talking about a triglyceride, you only need one double carbon bond to call this a unsaturated fat or triglyceride. Any question about any of that? And you can see the kink right there where the double carbon bond is. So if you had this fatty acid here instead of that one, this would be a saturated fat with one double carbon bond in the triglyceride. This is now an unsaturated fat. Unsaturated fats are better for the heart. Saturated fats are not as good for the heart. We'll just word it that way. And all you need is one to make this better for the heart. And why is this molecule is a little easier to bring into solution in the blood than a saturated fatty acid. And for some reason, this one, I'm not exactly sure why, when it's in the blood, it doesn't cause problems with the heart, whereas a saturated fat can, or is more likely to. Triglycerides are the body's richest energy source. They're stored as adipose in animals. You can just look down at your belly. Everyone's got a layer of fat underneath the skin and their belly. I suppose not somebody who's starving, but everybody else would have a layer of fat there. By mining the body's richest energy source, if you were in Point Barrow, Alaska in the wintertime and you only had a light jacket or no jacket at all, and it's freezing outside and your car breaks down, and you have 20 miles to walk into town, the best thing to do would be to grab some fat or triglycerides, as much as you can carry, because when you eat it, you would get the most energy per gram of fat than any other per gram of any other substance like protein or carbohydrate. You get more energy from fat. It wouldn't be necessarily the healthiest thing to eat, but since you need the energy because it's Point Barrow, Alaska in the wintertime, below freezing, I don't know what Point Barrow gets, but it's really cold. And if you don't know, their winter is the entire winter long. They never have the sun come up above the horizon. Any question about any of that? So you get the most energy from carrying fat because you get the most energy per gram of any other food substance. Any question about any of that? All right. As I mentioned, all but one of the lipids is insoluble in water. There is one lipid which is soluble in water, and it's the phospholipid. Phospholipids have two fatty acid tails, and yes, one is always saturated, the other unsaturated. Look to a glycerol right there. This is the glycerol. And in the third position, we don't have a fatty acid. We have a phosphate group, which will be linked to an organic group. The R stands for the organic group. The R can be anything organic. This region of the phospholipid, called the head, is actually polar from both the glycerol and the phosphate group, especially the phosphate group is polar. The glycerol, not so much. It's not really polar, meaning the carbon and hydrogen, the carbon and oxygen are not polar. But the phosphate group is polar. It's actually ionic. There's a complete -1 charge there. The fatty acid portion is nonpolar, and that makes the phospholipid being biphasic, with the heads being soluble in water, meaning polar, and then the tails being non-soluble in water or soluble in lipids like oil. By a biphasic molecule, it means it can be soluble in either water or lipids. There are not too many molecules like that. However, soaps and detergents are other biphasic molecules, and the reason why we add soap to our laundry when we setting the washing machine is so the water will dissolve the polar molecules in our clothes and pull them out to clean them. And then the soap will dissolve the lipids in our clothes. And that way, when we add the soap, it'll clean our clothes better and more thorough than just adding water alone. Or for that matter, adding oil. Nobody cleans their clothes in oil, but you could if you wanted to. Any question about any of that? So a biphasic molecule is soluble in both water and in lipids like oil. There's the phospholipid shown in a stick figure with the head that way, the tails this way, nonpolar. When we put phospholipid together in water, they start lining up like this or like that. This is the same, actually, just a different configuration instead of um, one saturated tail and one unsaturated tail in the fatty acid. This is all unsaturated fatty acids. Um, the point is when we put phospholipids in water, they start lining up like this, where the tails, which don't like to be around water, they like to be around other nonpolar regions, come together like that, and then the water will be on the outside here and here, and then the heads will be facing the water because they like to be around water. The tails will be on the inside because they don't like to be around water. And this is actually the way that the cell membrane is made up of. For animal cell membranes, we actually have cholesterol inside the membrane of our cells, inside between the phospholipid, and the cholesterol adds strength and support to the membrane or the phospholipids to make the membrane a little stronger and a little more held together. Any question about any of that? And although you first get the phospholipids showing like this when you put them in water, they will actually bend around and come like this, or maybe like that. And this one is important because on the inside we have water. Facing the water, we have one layer of phospholipid, the heads. Then we have the tails of the phospholipids together because they don't like to be around water. They like to be around other lipids like the tail of the phospholipid. And then on the outside, we have the heads because outside this membrane will be water, and the heads like to be around water. What does this remind you of? This shape remind you of anything?
A cell.
Yeah, this reminds you of a cell composed of phospholipid being the main molecule in the cell membrane. On the inside of the cell, we have the heads of the phospholipid bilayer facing the inside because the inside is mostly water. The inside of the cell is cytoplasm, which is mostly water. And then outside the cell, we have mostly water. Doesn't matter where you are, even in the your bone, the majority of the bone matrix is water. But let's talk about the blood. Everyone can agree this is a cell in the blood, and what's going to be on the outside is other blood, which is mostly water. Everyone agrees with that. And so we have the heads facing the outside of the cell, which is also water over here outside the cell. Any question about any of that? All right. So that's fatty acid, simple lipids like triglycerides, phospholipids. Let's now move on to our third group of lipids, and that's a steroid. A steroid is four interconnected rigid carbon rings, characteristic of the group 1, 2, 3, 4. At each point of the group, we have a carbon, and if ever there's no carbon in this point, you have to literally put in the atom. Let me show you that if I can. What slide am I on, by the way? Because I have to remember. 30. I don't have one. Let's go to glucose. Pull that up so you can see it. So right there, we have an oxygen because that point is not a carbon. Same with this position right there. Wherever it's a carbon, you just leave it a point. And where it's not a carbon, you have to put in the atom that is in that position. Any question about any of that? So this is a steroid. If ever we have an OH group attached to a steroid, we call it a sterol. Cholesterol has one OH group on it, and so this is a sterol. Cholesterol is important in our cell membranes and in our body. In our cell membranes, cholesterol gets in between the phospholipids to give strength and support to the cell membrane, making the cell membrane stronger and having more support. Any question about any of that? If you don't know, you only find cholesterol in an animal product. You never find cholesterol in a plant product, like any plant or vegetable. You do not find cholesterol in soybean or soy products. Or do you find cholesterol in any bacteria? You only find it really in plants. Excuse me, animals. You only find cholesterol in animals. All right, any question about lipids? Oh, let me state one other thing. I did briefly talk about it, but um, the phospholipids are made up of monomers, and that is two fatty acids, a glycerol monomer, and then a phosphate group. So there's three different monomers making up the phospholipid. The triglycerides are always made up of three fatty acid molecules and one glycerol, and all fats are made up of fatty acids and glycerol. And these, the triglycerides and simple lipids, as well as the phospholipids, are made up of monomers, but not all lipids are made up of monomers. For example, cholesterol and other steroids and sterols are not made up of monomers. Any question about any of that? All right, if not, let's move on to the third group of the biological molecules, the proteins. Proteins are essential in cell structure and function. Proteins may be a structural protein found in bone, muscle, or hair. They may be an enzyme which helps speeds up a chemical reaction. They may be a transport protein which moves a molecule across the cell membrane or another membrane. They may be a regulatory protein that helps regulate the body functions like a hormone, like a child's growth hormone causes the child to grow. The protein could be responsible for movement. The reason why I'm moving my arm here, or the reason why we move, is because of contraction of a protein. This is also true for bacteria that can move. It's because the bacteria have a at least one flagellum, one or more flagella, which causes the cell to move. Proteins can be responsible for some bacterial toxins, like Corynebacterium diphtherium toxin is a protein. So is the poison on newts. It's a protein that the newt secretes on its skin. Proteins can also be antibodies. These are proteins that help the body defend itself against a foreign invasion, meaning antibodies can bind to and attack a molecule that's foreign to the body, including a molecule on a tissue that is foreign to the body. Anyways, it's telling you that proteins play a large number of essential critical structure and function to cells, and that they are the most diverse of all the biological molecules. Why are they the most diverse of all the biological molecules? Because instead of being made up of just a few monomers, proteins are made up generally of 20 different monomers. Many humans and most other organisms have their proteins made up of 20 different amino acids. For a carbohydrate, even if it isn't starch, which is only made up of glucose, it's usually only made up of a few different monomers. I don't know, maybe a dozen different monomers to make the sugars, less than 20. And I already mentioned lipids can be made up of monomers, depending on what the lipid is. Proteins are always made up of monomers that we call amino acids, and there are 20 different amino acids that can be in a position on a protein. You should know the general structure of an amino acid. Where an amino acid is shown here, there's always a central carbon and a hydrogen connected to that central carbon. There's also an R group An R is just an organic group. It could be anything. We'll show you a few different R groups in just a minute. In one end of the central carbon, it has an amino group, NH2. On the other side of the molecule, it has a carboxyl group, COOH. For example, here's tyrosine here, showing the R group there, the amino group there, and then the carboxyl group here. So this is the complete amino acid. Remember, proteins are just many amino acids linked together to form the protein. And before the last amino acid is added, you either call it a growing protein or a polypeptide because it is not a complete protein until after you've added the last amino acid. Any question about any of that? All right, here we're looking at different amino acids to a protein. Tyrosine having this R group attached in this position, the R position of the amino acid. And there are 20 different R groups for humans because there's 20 different amino acids. You don't need to know tyrosine, I'm just showing it to you. I'll mention something else. Here are some other amino acids. Remember, there's 20 different of them. Glycine, which is the simplest amino acid, it simply has a hydrogen atom taking the R group position. Alanine has a CH3 group. Valine, a 3-branch group of carbon and hydrogen. All of these R groups here, 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 and here are nonpolar R groups. But remember, every amino acid is actually both polar and nonpolar. The reason why it's nonpolar is the amino group and the carboxyl group are always polar, and you always have an amino acid having an amino group and a carboxyl group. And then some of the amino acids have a non-polar R group, and then other amino acids have a polar R group. So glycine, alanine, valine, leucine, isoleucine are all non-polar amino acids because their R group is non-polar. If you didn't learn from an earlier, I think it was an in-class activity, the polar group is determined by oxygen and nitrogen binding to hydrogen. The oxygen and nitrogen pull on the electron more strongly, so that electron, instead of being shared between two atoms, will be pulled towards the electromagnetically negative atom, in this case, oxygen or nitrogen, making a polar group. All of these are nonpolar because there is no oxygen or nitrogen in the R group. But that doesn't mean you can't have a polar R group. For tyrosine, this region of the R group is nonpolar, but this region is polar. You only need a little bit of polarity to make the R group polar. So tyrosine is considered a polar amino acid. Same with histidine. You can have the polar regions offside of the molecule. It will always be on the amino group, the amine group on one side, the carboxyl group on another side, and therefore every amino acid is always polar. But some amino acids are also biphasic because they are also, besides being polar, They are nonpolar. Phenylalanine, all the ones I showed you in the previous slide, are nonpolar. Whoops. And you only need a little region to be polar to make that R group polar. So this OH and tyrosine makes this R group polar. The same with this NH on histidine. This region is polar. Oh, that's nonpolar R group. That's another nonpolar R group. All right, any question about any of these? You do not need to know the different amino acids. You only need to know the general structure of an amino acid shown in this slide here. All right, amino acids are linked together by a peptide bond, one amino acid hooked to another, forming a chain of amino acids, eventually making the finished protein. How you link two monomers of amino acids together to make actually a dimer of a amino acid is you do dehydration synthesis, pulling out the OH group from one monomer, an H group from the other monomer. Let me blow that up. So now this N group here binds directly to that carbon group. And there it is here. This carbon group is going to form a covalent chemical bond to the nitrogen right there. This is a rare bond in the universe, as well as fairly rare on the planet Earth. You only see it really in the biological molecules that have a amino acid like a protein. Is it found in any other molecule? I don't think so. And that's why we call it a peptide bond, because it is found in peptides. Remember, a peptide is just a molecule shorter than a protein. When you're making the protein, you can call it a growing protein or unfinished protein. You can't say it's a protein until the last amino acid is added. Anyways, the point being that the peptide bond is rare in nature, but you do find it in proteins or peptides, and why it's called a peptide bond. And it's rare in the universe as well as rare on the planet Earth. Any question about any of that? To get the finished protein, you have to add all the amino acids, and after the last amino acid is added, you now have the protein, which I call the finished protein, just to make it sure you don't get confused. But that's actually the protein. And before that point, you can call it a growing protein, or you can call it a polypeptide, meaning a shorter molecule than the protein. Any question about any of that? All right, let's talk about the levels of a protein structure. You can have 4 different levels of protein structure. The primary level, the secondary level, the tertiary level, and all proteins have the primary, the secondary, and the tertiary level of structure. Some proteins have a quaternary level of structure. Any question about any of that? If not, let's watch the video on the 4 levels of protein structure. I told you my computer was slow.
Most people know Grammarly as a spelling and grammar tool, but it's actually so much
After
more
the commercial.
AI writing assistant that works wherever you work so that you can craft
this video, we're going to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many...
Okay, I'm going to skip ahead to the different levels of structure.
You're forming a covalent bond, which is hard to break. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's two of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you can see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the three-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we can see some areas it's just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, let me state this video was a little bit of an oversimplification. Oh, let's see if I can get to it.
In this lesson, we're going to talk about...
In the secondary level, it was talking about that the hydrogen bonds stabilize. I'm not even finding it. Oh, this is the wrong video, that's why.
In this video, we're going to talk...
There we go. It said that the hydrogen bonds stabilize the structure of the secondary structure. There are other bonds as well as repulsions that help stabilize the secondary structure. They, in this video, are only talking about the major bond, which is the hydrogen bond, but there are other forces of attraction and forces of repulsion that make the secondary structure. And I'll talk a little bit about those. Anyone got any questions about this video? Close this. Oh, come on. All right. The primary structure is simply the specific sequence of amino acids in a polypeptide chain. So once the protein is finally made, we have the primary structure, and that's simply the sequence of amino acids one after the other. Once the protein is made, the protein then starts interacting with each other. And what happens is that the nearby amino acids start interacting with each other. And that we call the secondary structure. The secondary structure is always localized interactions. between neighboring amino acids, meaning you do not have amino acids far away from each other interacting with each other. You only have neighboring amino acids interacting with a neighbor amino acid. These interactions can be either from attraction, forces of attraction, or forces of repulsion. And that makes the secondary structure. There's actually three secondary structure, the alpha helix shown here, the beta pleated sheet shown here, and then the random coil, which they didn't talk about in the video. Let me see if I can find it. Where is paint? There it is. A random coil is just where a protein moves at random. Bring it out, bring it back in. So here's the first amino acid. Come on, mouse. First amino acid right here. And it didn't work. And there's the last amino acid right here. And along the way, we have different amino acids. And this is actually the tertiary structure because they're not nearby. But my point is that they can interact to get to the random coil. All right, let's go back to the secondary structure. The secondary structure can happen when nearby amino acids start interacting with each other. Oh, come on. I'm trying to blow this up. Here we have two amino acids, well, let's go there, that are interacting with each other. And this shows you a dotted line. That means a hydrogen bond or some other bond is attracting these together. And I guess it has to be a hydrogen bond because that's what a dotted line stands for. But there can be other forces of attraction. For example, between this one, if this amino acid has a positive charge and that one a negative charge, these two amino acids will be attracted to each other. A positive and a negative complete charge that makes an ionic attraction between these two amino acids. You can also have another force of attraction, and that is van der Waals forces, which two nonpolar amino acids will like to be around each other and they will be attracted together. An example of that would be your bottle of liquid olive oil. The different olive oil molecules are attracted to each other by van der Waals forces, and that attraction is strong enough that the liquid molecules of olive oil can be attracted to each other by van der Waals forces, making the molecules act like a lipid, where the liquid, sorry, not lipid, act like a liquid where the molecules are attracted to each other and then form, and they break all the time, similar to the hydrogen bonds in water. But the van der Waals forces or forces of attraction between the olive oil molecules, the polar molecules are attracted to each other by van der Waals forces, making the olive oil in a liquid. And that's why we have liquid olive oil. If we did not have the van der Waals forces and the olive oil were not attracted to each other, the olive oil would be gaseous olive oil. If ever you have it so that the van der Waals forces never break, you will then have solid olive oil. And if you put olive oil in the freezer or even the refrigerator, the olive oil will become solid. And that's because the energy of the molecules or the temperature of the molecule, it's the same thing, they are moving slow enough around that they cannot break the van der Waals forces. And then the liquid olive oil will be a solid. My point is that nonpolar molecules can be attracted to each other, forming a force of attraction. You can also have forces of repulsion, like these are held together by nonpolar attraction, van der Waals forces, this one by an ionic force of attraction, that one by a hydrogen bond. You can also have forces of repulsion. If this one has a positive charge, actually this one has a positive charge, and that one has a positive charge, they will be repulsed from each other. Two positive charges are not going to want to be by each other. They'll be repulsed from each other. You can also have a repulsion, and that is if this is an ionic molecule or a polar molecule, and this is a nonpolar molecule or monomer, it will be repulsed from that polar monomer. That's a force of repulsion. Any question about the different forces of attraction and repulsion that we have playing out in the secondary level of protein structure? Remember, you only have the secondary structure between neighboring amino acids. So all the amino acids start interacting are nearby each other, they're neighbors. They don't have to be immediately by each other. Like this one is what? 1, 2, 3, 4, 5 away from that one. The tertiary structure is the overall three-dimensional structure of the polypeptide chain, where we have all the forces of attraction and repulsion that we saw in the secondary structure. Then you can have hydrogen bonds, you can have ionic bonds, you can have ionic forces of repulsion, you can have nonpolar and polar molecules being repulsed from each other. You can have van der Waals forces of attraction. You're going to have all of these same forces of attraction in the secondary structure as you have in the tertiary structure. The difference is that in the secondary structure, this only happens between neighboring amino acid. In the tertiary structure, it can be any two amino acid, can be repulsed or attracted to any other amino acid, meaning that this tertiary structure happens all over the protein. In this one example, we have a force of attraction between the last amino acid of the protein and one of the early amino acids of the protein. There's one other difference in the tertiary structure, and that is we have a force of attraction in the tertiary structure that you do not see in the secondary structure, and that is a disulfide bond. Let me get out paint again here. Right here. No, I probably have it a little too close together, but we can have, let me change this, bring it, link it like that. I need a little bit more room, so I don't want it being real close. All right, we can have a cysteine amino acid right here. So this is cysteine, which has a sulfhydryl group right here. and a hydrogen sulfur group right here. And the cysteine is right here. Let me bring this over here. So the sulfhydryl group is part of the cysteine, combined to the cysteine. And then we have another cysteine right here. And I've got the sulfhydryl drawn in already. Oops, in the wrong place. Let me see if I can move that up. I'm not, so there it is. Just want to move that up a little. It'll break that protein, but not terribly important. Move that down a little. Oop, come on. Oh well, just leaving it. All right, the hydrogens can come off. They go into solution, and that will allow the sulfur to form a covalent bond between each sulfur. And this is a chemical, very strong bond. You actually get a sulfhydryl bond made when you perm your hair, and that's why you get the kink in your hair of the perm from this sulfur dibridge. I mean, the sulfhydryl comes off, forming a sulfur link of the cysteine, where cysteine is linked, cysteine to cysteine, by this sulfur dibridge. Any question about any of that? So that's a real strong link. The point is this amino acid here can bind to this amino acid here. And that is called the disulfide bond between two cysteines. Any question about any of that? All right. For many proteins, you can only get up to a tertiary level of the protein structure. Like for all enzymes that I know of, catecholase, catalase, sucrase, there's only one polypeptide unit making up the protein. So these can only go up to a tertiary structure. But if there's more than one polypeptide making the protein, you can then get a quaternary level to the protein. So only some proteins have a quaternary level, and these are the proteins made up of more than one protein unit, meaning more than one polypeptide unit making up the final protein. What happens here is you have forces of attraction and repulsion between the different protein units, polypeptides. An example of that they talked about was hemoglobin, as well as antibodies, where the different protein polypeptides come together to make the finalized protein, and that's called the quaternary level. Any question about any of that? Hemoglobin and antibodies are made up of 4 polypeptide units to make the complete protein, which we'd call the antibody or the hemoglobin molecule. Proteins, you can classify them by their shape. They may be globular, meaning roughly compact, roughly spherical. Hemoglobin is globular protein, or the protein can be fibrous or more thread-like. Collagen is a fibrous protein. You can also have conjugated proteins. These are a combination of a protein or amino acid subunit with other organic or inorganic components. These conjugated proteins are named by their non-protein component. Like a glycoprotein is a protein added onto a glycogen, or glycan. A lipoprotein is a protein combined to a lipid, and you can find a lipid protein molecule in your blood. You can find glycoproteins in your blood and on your cell surfaces too. Any question about any of that? Proteins can denature. When a protein starts to lose its three-dimensional shape, such as by breaking hydrogen bonds, like when you're heating the protein up, or forming hydrogen bonds when you're cooling the protein down, like putting it in the freezer, you can get the protein start losing its three-dimensional shape. A protein that is no longer functional is said to be totally denatured. You can denature a protein by putting it in the incorrect temperature, the incorrect pH, or the incorrect salt concentration. Any question about any of that? All right, let's talk about the fourth biological molecule, the nucleic acids. There are two you need to know about, DNA and RNA, which are also called DNA for deoxyribonucleic acid and RNA ribonucleic acid. All of these, meaning DNA and RNA, consist of a monomer subunit called a nucleotide. Nucleotides consist of three parts, a 5-pentose sugar. Let me blow this up. So this is a nucleotide here containing a 5-pentose sugar, meaning a 5-carbon sugar. A phosphate group shown here, and then a nitrogen-containing base shown here. This nitrogen-containing base can be a purine, shown here, or a pyrimidine, which will be shown here. This is another nucleotide right here. Any question about any of that? For DNA and RNA, there are only 4 nucleotides making up either the DNA or the RNA. Let me end this. For the purines, they are adenine or A and guanine or G. And these are 2 of the nucleotides you find in DNA. The other 2 nucleotides you find in D are cytosine and thymine. Cytosine or C, thymine or T. Cytosine and thymine are pyrimidines. How you can easily remember in DNA which nucleotides are pyrimidines is the word pyrimidine has a Y in it, and then cytosine and thymine have a Y in them. Adenine and guanine lack a Y, and purine lacks a Y. So for DNA, it's very easy to distinguish what's a pyrimidine and what's a purine. Now for RNA, we have also 4 nucleotides. 3 of them are the same as DNA, and that would be the A, the G, and the C. These are both in DNA and RNA. But RNA does not have T or thymine. It has uracil replacing the T in RNA. Uracil does not have a Y in it, but uracil is a pyrimidine. And how you can remember that uracil is pyrimidine is uracil replaces thymine in RNA, and thymine has a Y in it, so uracil and thymine are pyrimidines. Any question about any of that? All right, the DNA bases bind to each other by hydrogen bonds, and they form complementary base pairing, meaning the A and the T always hydrogen bond with each other. And so the A will have a complement on one strand of DNA, which will always be T on the other strand of DNA. because the A and the T hydrogen bond to each other. And then the C and the G are complementary, meaning on one strand of DNA, you'll have a C. On the opposite strand of DNA, you'll have a G. And that's because the C and the G can form hydrogen bond between each other. The hydrogen bonds between A and the T are always two hydrogen bonds. And between the C and the G, they are always three hydrogen bonds. Remember DNA, the two strands of DNA are only held together by the hydrogen bonds between the strands, the nucleotides of the strand of DNA. One strand of DNA is held together by a chemical covalent bond holding the DNA together. So that's a strong bond holding the single strand of DNA together. The double strand is only held together by the hydrogen bonds between the complementary bases. And that's a weak bond holding the two strands of DNA together. Any question about any of that? Let's talk a little bit more about DNA. It is made up of sugar, and it's always deoxyribose right there. This DNA is normally double-stranded in your cells, meaning one strand of DNA will hydrogen bond to a second strand of DNA, and normally that's the way your cells are. However, your cells can have single-stranded DNA whenever the DNA is replicating or whenever the DNA is being transcribed. You'll have a region of the DNA which is single-stranded. And the region will be the region actively undergoing either replication or transcription. The DNA single strand is held together by the sugar phosphate backbone. Let me see if I can blow this up and show you. So here we have a sugar, the green, the phosphate being the orange, held together by the next sugar, held together by the next phosphate, and that holds the single strand of DNA together. The bases are like steps on this helical staircase, and the bases hydrogen bond with each other, binding the one strand of DNA to the other strand of DNA. So DNA actually looks like a twisted ladder or a spiral staircase, with the bases being the rungs on the ladder. Any question about any of that? RNA has the sugar ribose, which is also a 5-carbon sugar shown right here. It is always single-stranded in our cells, unless you have a virus which is double-stranded. In your cells, you'll only have RNA which is single-stranded. It is held together by the sugar phosphate backbone, holding the RNA single-stranded RNA molecule together, and that's a chemical covalent bond, strong bond. There are different bases, or at least one different base from DNA, and that is uracil replaces thymine. There are 3 major kinds of RNA: transfer RNA, called tRNA, also called tRNA, messenger RNA, also called mRNA, and ribosomal RNA, also called rRNA. All 3 of these RNAs are important in protein translation. Any question about any of that? All right, I'm going to stop here. There are 3 more slides I need to talk about in this lesson. I was hoping to finish this lesson, but I didn't quite make it. Any questions before I go? Oh, there's a chat there. Remember, if you have a chat, I'm not going to see it. All right. All right.